Assalamu alaikum. Welcome to my lecture. In this lecture today, we are going to discuss about the levels of the structural organization of proteins. Proteins are the vital for the normal cell function. These proteins are made up of the very long chain of the individual units. So individual units in case of proteins, these are the amino acids. These amino acids are joined together by the peptide bond. So when these are joined together by the peptide bond, they form a amino acid chain. Then they have sort of, you can say they have sort of resemblance with the strings and the beads that are twisted and folded into the final protein shape. So, actually, in order to make a protein, we need to get to know two things. First are the ingredients, and second is the recipe. So, here, the ingredients are these amino acids that are the basic units of the proteins. So, these are the ingredients for making protein, and how the finished amino acid chains basically folds into the proteins. This is the recipe that we need to know about making proteins. So what happens? Humans use 20 amino acids in our day-to-day routine for making proteins. Now, let's talk about their structural organization. Proteins have multiple levels of the structural organization. These include their primary structure, their secondary structure, and then its tertiary structure and quaternary structure. So this is the primary structure of the protein. All these levels, like primary structure, secondary structure, tertiary, and quaternary, they basically create a hierarchy. You can think as an analogy. For this, you have to think about the alphabets. So here you can see these alphabets can be used to create words. Okay, these alphabets can be used to create words which can make simple sentences, and that can further be made into the complex sentences. As here you can see, these are simply letters, these are simply alphabets. This A, U, V, R. So what these letters basically themselves would be considered as the primary structure. So primary structures are considered as simple alphabets, and these simple alphabets are used to create the words. So simple words here, they would represent the secondary structure. For example, here you can see the words antibodies, useful, science, tool, vital. So all these words basically, if we think the hierarchy of the protein as the paper, as the alphabets, then here you can see the simple letters would represent themselves as the primary structure, and when these letters are used to make words, then these letters would represent the secondary structure. Now the tertiary structure would be when entire chain gets folded together. That may be, you can say, making a simple sentence. So here you can see these are the simple sentences like antibodies are a useful tool, science is a vital. So these sentences basically would categorize themselves as the tertiary structure. Now, quaternary structure, actually, you can say that can be a few polypeptide chains when coming together to form more complex proteins. So these, in terms of alphabets, these are to make more complex sentences. As for example, antibodies are useful tool in the advancement of the research, then routine practices in the science. For example, Western blot, they basically rely on the antibodies. So what happened here, from simple letters to complete complex sentences, there are four different levels. So simple letters basically represents the primary structure of the protein, as here you can see, and then they converted into the words that are basically the secondary structure of the protein, and then the simple sentences would represent themselves as the tertiary structure, and the complex sentences would be the quaternary structure of the protein. When it comes to the primary structure of the protein. So primary structure here you can see is the simply the linear sequence of the amino acids. So these amino acids are simply connected through the peptide bonds. So you can think example like strings and pearls. So peptide bonds between the amino acids, these are basically very rigid bonds. So here you can see in the red, these are the simple units of the proteins. These are simply amino acids that are linked together through the peptide bonds. Okay, so these peptide bonds basically holds themselves together. So this is simply the primary structure of the protein. So as I told you that these amino acids are basically very rigid, but when by comparison these single bonds, when connecting the amide functional group of the peptide bonds to the alpha carbon, so these are basically flexible, that allows the significant freedom of rotation. What does that mean? That means this polypeptide has a significant freedom to rotate. It is able to rotate. Okay? And through that rotation, basically protein can fold into one of the two types of the secondary structure. So here the bonds are itself, they are rigid, but when they link to the alpha carbons on the both sides of the peptide bond, then they basically make able this particular structure to rotate. So they have freedom of rotation. So if we talk about their functionality, then normally the primary structure of the protein is basically non-functional. You can take an example of the insulin. So insulin is basically a protein, but it acts like a hormone. So what happens here? Insulin is, you can say, this is an exception for the primary structure of the protein, for the primary level of organization of the protein. As I told you that the primary structures are normally non-functional. So these are the very first structures that are formed after the process of the translation. So these are basically non-functional. There are several modifications that they have to gone through in order to make them functional. So one exception is there, that is the insulin. So insulin is the has the primary structure, but it is specifically functional protein. Now, when it comes to the secondary structure, as I told you that this primary structure has freedom of the rotation. So what happened here? This primary structure will, you can say, can fold into one of the two types of the secondary structure. So these are the two types of the secondary structure. These are the alpha helix and these are the beta pleated sheets. So these are the two forms of the uh, secondary structure of the proteins. So here you can take example that this alpha helix basically resembles a spring. So this helical structure brings the CO of the first amino acid near the NH of the fifth amino acid. You know there is also a bond. Let's say there is one amino acid that is present here. Okay. So let me draw. So this is the one amino acid that is present here and other amino acid that is present here. So you can see that this helical structure brings this CO of the first amino acid near the NH of the fifth amino acid, and the second CO gets near the sixth amino acid, and so on. In simple words, you can say each of these instances, these are separated by the four amino acids. Four amino acids जो हैं, they are between these two amino acids that are basically making the bond. So here you can see the bond, there is a bond formation between these two amino acids. So this particular bond is the hydrogen bond. So this hydrogen bond is between the first amino acid and the fifth amino acids, and between there are four amino acids. So this helical structure you know uh, these are really stable because of the hydrogen bonds. So when we talk about the beta pleated sheets, they also rely on the hydrogen bonding. There are also hydrogen bonds that are holding the pleated sheets together, that are providing them the stability. So you can think this as a for example, there's one flap of the paper and the CO on the another flap of the paper. Okay, so we have two flaps on the paper. So one flap is having the particular high CO group, and another flap of the paper having the NH group. So a hydrogen bond is basically forming. So these bonds almost hold the pleated sheets together. Okay, so these, because of these bonding, the beta pleated sheets are, these are really stable as well. Now the tertiary structure is here. You can see the example of the beta pleated sheets is the silk fiber. So, and the alpha helical structure is the keratin. Keratin of your hairs, your nails, that basically provides support and shape to your nails and hairs as well. So here you can see the difference between both structure is that in alpha helix, as it is the helical structure, there is a bond formation between the single amino acids of the single chain. ठीक है, यहाँ पे single chain है that is folded in the form of a helical structure. So there is a bond formation between the two amino acids within the single chain. Here we have two chains, and one hydrogen bond formation is between the one amino acid of the one sheet and another amino acid of the other sheet. So these are basically the secondary structures of the protein. So hydrogen bonding of the, you can say, peptide backbone, that basically causes the amino acids to fold in a repeating pattern. So that basically form the secondary structures in the form of the helix or the sheets. So as I told you that these are the simple words that are produced from the letters. When it comes to the tertiary structure of the proteins, so these tertiary structures, now what happens, tertiary structure is that this is the overall shape of the polypeptide chain, that basically includes the secondary structures as well as the other features. For example, let's say this is the second tertiary structure of the proteins. So this secondary structure is also, you can say, it's specifically functional at the various sites. So here, though, whenever any substrate has to come and it has to interact with this particular structure, then this particular structure has specifically the specified functional groups are there that can interact with the coming substrate. So you can think the tertiary structure, for example, enzymes, they have specific region, that is, you can say active region, specific catalytic region, where these are going to perform their catalytic function. So these particular sites are basically the active sites. So these are those sites in the, you can say, in the tertiary structure that are specifically functional. So there are functional domains, there are functional regions, functional motifs in the tertiary structure of the protein that have specific functional groups that are present there in order to interact with the upcoming molecule or upcoming substrate. So for example, enzymes basically comes under the category of the tertiary structure of the protein. So basically, tertiary structure is the three-dimensional folding pattern of the protein because of the side chain interactions. These side chain interactions are basically those, let's say, we have, where is the pencil? Okay, so let's say we have two sides here. So one is here and one is here. So these particular sites are having specific functional group. Let's say OH functional group is here, and the, let's say this OH functional group is here, and then we have amide function, amino function, NH functional group is here. So whenever any substrate is going to come here, it will come here, and then it will bind and interact with these sites that are basically the specific domains for the interaction with the other sites. So these are basically the specific functional domains. So you can say that the tertiary structure of the enzyme is basically the specifically functional because of these side chain interactions. When so you can say that the secondary structure basically includes the tertiary structure basically includes the secondary structures, including beta pleated sheets, including alpha helix, as well as the other features. You can say when the two sulfur containing amino acids such as cysteines, they can bind here to form the disulfide bridges. For example, because these both these amino acids having the cysteine, basically these are the cysteine residue that have the sulfide that have containing the sulfur, so they can form the disulfide bonds. So and also the hydrophobic amino acids, they form the bonds with the, you can say, one another. And in case of hydrophobic amino acids क्या करते हैं, they basically orient themselves towards the inside of the proteins. So in that way they basically avoid the contact with the water, as these are the hydrophobic amino acids. Now, tertiary structure, you can think that tertiary structure is a kind of a like way when your headphones get tangled up in your pocket. So when it comes to the quaternary structure, then quaternary structure is basically the final level. It is the final level in the structural organization of the protein. So this quaternary structure is that in which the multiple polypeptide chains basically come together, they to form the larger proteins, such as these basically includes the hemoglobin, which is a very important transport protein in our blood. So if we talk about the hemoglobin, then the globin part of that hemoglobin basically made up of the four chains, in which are the two alpha chains and the two beta chains. So this is the classical example of the quaternary structure of the protein. So these refers to as those proteins that containing of the two or more than, you can say, more than two amino acid chains or polypeptide chains. So these are the you can say the quaternary structure of the protein. So here you can see that the four polypeptide subunits, for example, two alpha subunits and the two beta subunits, so they basically come together, and that have come together, so they are to form a single hemoglobin molecule, hemoglobin protein, which is roughly, you can say, it's a tetrahedral arrangement of all the polypeptide chains. Now, there are several, you can say, several forces that basically are present in order to stabilize those structures. So when it comes to the primary structure, then the force that is going to stabilize this particular structure is the peptide bond. So these are the basically bonds that are present between the uh, amino acids. Amino acids are basically linked together, joined together with each other with this peptide bond. And when it comes to the secondary structure, here the stabilizing force of course it is the hydrogen bond. So peptide bond is always there, as it is the sequence, or it is the amino acid that are linked together by the peptide bond. And then these are the hydrogen bonds that are going to stabilize these sheets as well as helix. So what's the difference here? In the alpha helix, the hydrogen bonding is between the amino acids of the single chain, ठीक है, and in the center there are the four amino acids. And in case of beta pleated sheets, these hydrogen bonds are formed between the two chains, one amino acid of the one sheet and other amino acid of the other sheet. And the other forces that are basically important for stabilizing the tertiary structure, these are the peptide bond, of course, then hydrogen bonding, and also there are other other interactions like covalent or ionic bonds, ionic forces. And then in case of sulfur containing amino acids, there are disulfide bridges and other weak interactions like the van der Waals interactions. Van der Waals forces are there in case of the tertiary structure. So all these forces forces are basically there to stabilize the particular tertiary structure. And then it's the primary structure. So here again there are all the forces that are present here, as these are the more than two polypeptide chains that are joined together to form the particular functional molecule, functional protein, in fully you can say the fully folded structure of the protein, that is the quaternary structure. So here the forces that are responsible for their stability, these are the peptide bonds and then hydrogen bonding, as well as the ionic or covalent interactions, and in case of sulfur containing amino acids, these are the disulfide bonds and the van der Waals interaction. So this is all about the today's lecture. Today we discussed the simply overview of the hierarchical, you can say, organization of the protein. So if you like the video, then don't forget to subscribe my channel, and thank you very much. Allah Hafiz.